Great. Thank you, Mina, for the invitation. Are you looking at my screen now?
Yes, I
Excellent.
can
That's good.
definitely see your cute kitty cat there.
All right, good. So I thought it would be quicker to say what hasn't happened in 2021. It would be a shorter talk. So we'll see if we can't get through this quickly. Actually, it's been a very busy year in terms of diseases, and I think all of you have noticed that too. The main thing that I'm aware of is that Phytophthora was a much bigger player than it usually is. I find that often people are really worried about Phytophthora, but it doesn't always show up. This year it did show up, both in nursery and landscape situations. And as you probably know, the Phytophthora is not really a fungus. It's an oomycete, or we would call it a water mold to be a little more familiar. And it has a life cycle that's very much tied to water. So in order to make its zoospores swim out into new areas to do new infections, it requires a wet environment. So the more it rains, the happier it is. And as you probably noticed, this was an unusual growing season on Long Island because we didn't get our midsummer drought that we're used to having. So there were periodic rains through the entire growing season, and that created a wet situation at just about every possible temperature point. So every disease found its moment. And in particular, Phytophthora were quite enthusiastic this year, I would say. Hold on here just a second. I have to get my, there. Here's one of the things that Phytophthora can do. It will attack the European beech, Fagus sylvatica, and cause a canker disease that's referred to as a bleeding canker because it can actually look rather grisly. This is a really good time to do this talk because we're close to Halloween. You see those very wet areas. You'll have a reddish brown stain coming out of areas that are infected by the Phytophthora. And it's actually not just one organism that does this. There are at least 3 species that have been named as affecting the beech in North America, and I think that list will just grow. The organisms can be splashed up from the soil into the trunk, and then it's possible for the Phytophthora to spread through the xylem and move up the tree. So you can actually see the bleeding cankers fairly high up the trunk of a beech tree. Fortunately, with this one, we've learned that we can work with a phosphite type material, a phosphorus acid material, along with some pentrabark, and do direct treatments on the trunk and really help these old trees survive in spite of this attack. There are other things that Phytophthora can do too. One of the most common is a root rot during some sort of container production. Here you see some Rudbeckias that look like they have really quit trying to grow. And that's because of a Phytophthora infection, which is much more obvious once you look into the pot, and you can see this very dark brown discoloration and a softening of the outer tissues of the root. You can't tell the Phytophthora root rot from a Pythium root rot just looking at it, but there's something about the speed and the the aggressiveness of Phytophthora that makes it often a worse disease to have than a Pythium problem. We had a number of calls on skimmia in the environment this year, and here's an example of a plant that had wilted down after a fairly recent planting in the landscape. And one of the things you'll see with this plant is a good illustration of the crown rot capacity of a Phytophthora. A crown rot means that it's the root crown that's showing symptoms. So you might also have a root infection, but you can see the browning coming up into the base of the stem here, where the Phytophthora is growing, and we can isolate from this area to confirm what the pathogen is. Here you see a lavender with pretty much the same thing. It's a crown rot here too. When lavender gets sick from anything, it looks the same. Even botrytis can cause a very similar gray, dead look on part of the plant. Rhizoctonia can cause the same look. In this instance, though, it's a Phytophthora. Here you see a Stachys plant with the same sort of thing. Root rot moving up into the stem and causing a crown rot. Probably the most common problem this season was to see real losses in rhododendron plantings in the landscape. Usually our Phytophthora problems are seen in the first couple of seasons after an outplanting. I believe this was a fairly fresh planting in this case too, and you can see just destruction all the way down that line of plants. You can also envision that perhaps someone had supplied extra irrigation to hit onto those plants because they were recently planted, and the wetter it was, the worse it would have been for the plants in the presence of the inoculum. Here's a close-up showing you how the plants have really defoliated. You can see some brown areas in the foliage there, but basically you're looking at a vascular wilt kind of problem for these plants. The root system's probably rotted, and then the xylem has been invaded by the Phytophthora. If you look closely at these leaves, though, you see something that we don't see every year. This was just from a outdoor grown plant, and they're large, irregular brown lesions, darker at the outer part of the lesion, which are being produced by a Phytophthora that's a foliar blight. There can be both the root attack moving up into the plant as well as a foliar blight. Sometimes something like Phytophthora cactorum can do both, but there's some Phytophtheras that are specialized in one or the other of those kinds of attack. Here's a little bit more zoomed in, an opportunity to see one of the things that I look for. If you notice over here, there's a petiole on the left, lower left, there's a petiole that's brown, and the browning is just bleeding into the leaf from the base. That is a very classic Phytophthora look. So I'm always going to be checking for the possibility of that if I see that kind of a symptom. It's often hard to tell Phytophthora from Botryosphaeria, but the conditions are different. Botryosphaeria is really favored by droughty conditions that stress the plant, whereas Phytophthora comes from lots of wetness and splashing. In particular, we've seen this foliar blight happen in garden centers or wherever you have a holding yard where there's some sort of a tall riser irrigating onto the top of the plant. So more water hitting the foliage is going to make this much more likely. Another plant that is vulnerable to Phytophthora is English ivy, and also other variants of ivy as well. When Phytophthora is attacking, rather than having small round spots, you're going to see large brown blotches in the foliage. And there was a very interesting case just last week in New York City, where there was a rooftop garden with sections of English ivy going out. There's one right front and center here that indicates the symptoms. And this turned out to be a Phytophthora attack. And it perhaps would not have happened in another year, but we had so much rain. And again, perhaps because it was a new installation, they over-irrigated in enthusiasm. You certainly wouldn't want to be spraying the foliage of English ivy with water, even if it was a hot situation, because you would be really making an invitation to Phytophthora problems. Oh, and then there's this new Cornell tree that they've invented that's called a pumpkin tree. And the pumpkin tree is also vulnerable to Phytophthora. We noticed that the fallen fruits on the ground will show the rotting and the discoloration that's associated with the Phytophthora rot. And the tree is a little bit mythical here, but the growers on Long Island who import pumpkins from Pennsylvania in particular found that they had a whole lot of shrinkage this year, meaning that a lot of the fruit that they brought in would rot before they were able to sell it, because there was so much moisture that went into the upstate area in New York and also into Pennsylvania that made a perfect environment for Phytophtheras. So if you want to fight back against these things, first of all, you pray for different weather, but with the shoot blight, the above ground part, you can try to prune out areas where you see the infection. You try to adjust irrigation so that you're not having water hitting onto the foliage directly at all. You can make fungicide treatments. If you learn you have the problem, then it might be worthwhile to treat in the future using things like mancozeb or chlorothalonil or Aliat. You can also work with Pagent or copper or azoxystrobin or Mural. The general idea is to treat in the springtime, starting as the new foliage is beginning to be produced at a 7 to 10 day interval 3 times. The root rot is a more common problem on Long Island. Definitely you want to make sure that you get a diagnosis so you know what you have. If it's Botryosphaeria canker, you'd use very different materials to fight back against it. You do need to throw away the problem because you can't cure plants once they have a Phytophthora problem. So you're going to discard those plants and perhaps also discard the soil around them, because what you plant in that spot next might have the same vulnerability and you might lose it as well. Certainly reduce the irrigation if you think that it's been a little bit hyper in that area. And it is possible to also add some fungicide into the situation, especially if the customer wants you to replant with the same plant, which is so often the case. So you can work with things like Pagent or Subdue Max or Aliat or one of Aliat's relatives that are salts of phosphorus acid before you bring in the new rhododendron planting, and you'll have a little bit better chance of getting those plants through. There are some rhododendron species that are resistant to Phytophthora. which I listed just because they were in the Cornell guidelines when I was looking through it today. But I don't hear them mentioned very often. So I think it would be an example of something where if any of you have good experience with particular rhododendron cultivars that do well where others have failed, maybe you should share that information with us and we can have a more updated list of things that are really common in the trade. So let's move from the water molds into the fungi, the true fungi, which includes.
Margie, I'm going to interrupt you for
Okay.
the first poll. So everyone, if you want to get to your computers, and I will find the poll I'm supposed to launch. Okay. All right. You should all be seeing it on your screen. And I'll leave it up there for a few seconds.
And I can read it in case it's small for some of you. Phytophthora diseases are favored by a susceptible plant, flooded soil, high rainfall, drought, or all but D.
Yeah. And like, as Margie suggested, if you have encountered rhododendron that stay alive and you know the cultivar, go ahead and put that in the Q&A. It's not only for questions, but if you have a comment, feel free to put that in the Q&A because it will be nice to know.
That'd be great. Thank you.
All right, so we're approaching 45 seconds. I think we're good, and I will end the poll. Poll is being ended. All right, and I'll share the results.
Those are all true. Probably even the drought part is true. We're really thinking of Phytophthora as being favored by wetness, but if you stress a plant by drought stress, then when it does rain, the Phytophthora has a great time with it. So the all but D answer is the official correct answer here. Okay, so this is my complications. I need to get rid of that. Okay, got rid of that. So let's move to the fungi. They can do root rots also, and they can do leaf spots as well. So they're just as versatile as the Phytophthora. One of the fungal root rots is called black root rot. And many of you may have learned to call it Calabiopsis root rot, but that's gone now because now we have to call it Berkeliomyces root rot. We did see that on Phlox variegata this year, which was a little bit unusual. I've seen it on a number of plants, but that was a new one for me. And under the microscope, it looks like it does at the left. When you're actually looking at a sick plant, it's going to look something like this. It'll be stunted. It might have a lot of yellowing or a lot of purpling in it. And you will see an irregular stand of plants. They won't all be the same size. So if you're suspicious of black root rot, examine the roots and you can bring a sample to the lab to be tested. One of the things that helps with this disease is that you can manage it to a large extent just with pH. Most of our Long Island pHs are low anyway, but if for some reason you have a foundation planting and it's got a high pH, that's going to make the plants more vulnerable to the possibility of Calabiopsis or Berkeliomyces root rot. And if you need to use fungicides for some reason, the thiophanate methyl fungicides are the strongest against this. And it's never good to use that group 1 fungicide alone. So you'd want to rotate it with a firm, for example, to have management. I've never known anyone to have to use fungicides in the landscape, however, so that's more of a production situation. So fungal leaf spots, however, include a lot of really interesting and zany things. These are pictures that Mina got this summer of a really intense and scary blighting on Acer rubrum in the landscape. And the lower foliage in particular was very much blighted and was defoliating. And this was at the end of July that we saw this. And it turned out to be a disease that I'm really not familiar with, even though I've heard of it before. We found that it would culture out very nicely and start growing sclerotia on the plate. It acts sort of strange as fungi go. And this turned out to be the Cristulariella leaf spot. They've renamed that fungus too. It's now a Grovesinia moricula, and it has many hosts. So I thought after we discovered this that the weather had been wet and we were going to see lots more of it on many other plants, but there was not a single other case of it the entire season. We saw plenty of symptoms on that first tree. They were down the veins and taking out large patches of leaf, as well as circular spots. But I checked with friends down in Georgia because I learned that pecan was one of the hosts of the disease, and learned from them that anytime they have a cool, wet summer, they're liable to have a real epidemic year for this problem. So maybe we were looking at the combination of our wetness along with a little warming of our climate that created this problem for us this year. What they have seen is spots on many different kinds of plants. On the left, you see grape with cristallariella leaf spot. On the right is a pecan leaf. And after I spoke to my pecan pathologist friend, I found out about 2 weeks later that they were having the same problem we were in their pecan. Just, it was a good cristallariella year for them too. Boxwood blight. We saw this year. We didn't see a huge amount of it, but we saw it. And that's because it has its own sweet spot of high 70s and lots of rain to get it happy. And we were probably below or above that most of the time. We did see a number of the other things that create symptoms that resemble boxwood blight. On the left here, you see the macrophoma leaf spot, and on the right, the Voluatella leaf blight. They can be easily confused if you don't have a microscope and some patience to try to separate out what's happening. And I would just encourage you to make sure you know that you have boxwood blight before you make boxwood blight level decisions, such as pulling all the plants out or perhaps putting them on a really eternal type of spray program to keep the plants alive. We did see some boxwood blight, and Karen and I are both part of this boxwood blight insight group that's trying to put together all the information we possibly can to help you fight back against boxwood blight. So if anyone needs help with that, contact one of us and we'll do what we can to give you guidance on the problem. One of the most interesting things that I saw this year was Pachysandra showing boxwood blight. We've been telling you this can happen for years, but here's a real live case of it. This one came through Hillary Jufer in Westchester County. And at the right, you can see the spores of the Calanectria that causes this fungal blight on boxwood. But look, here we have spots on Pachysandra. And just as other people had told me, it doesn't sporulate on those leaves. You have to have a leaf completely dead before you'll get the sporulation of the Calanectria fungus on a Pachysandra. So that was an interesting thing to see. And I wanted to make this point to you also, that when you're dealing with a boxwood planting, it does not have to be thrown away just because you see boxwood blight. There have been many efforts by arborists to salvage boxwood plantings that have been successful. It takes money and it takes time and it takes repeated efforts, but it can be done. So in this instance, you're looking at a little hedge of boxwood in May of 2019. And here you're looking at the same hedge on the 14th of August, and it's green. Because if you have a well-established planting, it can put out a lot of new growth in one season. So you just need your customer to be a little bit forgiving about raggedness for a year or two before it comes back to beauty. And you need to spray with fungicides to keep that foliage protected once you have boxwood blight in the vicinity of those plants. The basic ideas for boxwood blight management are to prune out the hot spots before you go to do control. And Jim Lamondia's research is telling us that you really shouldn't prune in wet weather, as we know for many diseases, but you also shouldn't prune in dry weather, because the way the boxwood blight fungus operates, the spores can be spread during dry conditions as well. They just become little dust particles. So if you have an outbreak, get it identified, and then I would say to use fungicide on the planting first before you go in and do any type of pruning out of affected areas. It'll be a lot safer for the planting. There will be less spread of the disease. You certainly want to make sure there's no direct irrigation. You want to use mulch very skillfully, because you can cover over the fallen leaves that have inoculum on them if you mulch lightly, but every year in the springtime. Try to have your shearing equipment on a property rather than taking it from one place to the next. Fungicide treatments have focused mostly on chlorothalonil and propiconazole, with the chlorothalonil being a contact material and the propiconazole being a systemic material against boxwood blight. And then the good news is that we have some resistant cultivars now, in particular the new gen boxwood that are coming out of Virginia. The Freedom and Independence cultivars are giving you a choice of things to put in if people are tired of boxwood blight management and want to put in a highly resistant cultivar. This was an interesting case that we saw this season where the leaves were absolutely reddened by something. And it turned out that the reddening was from a boxwood leaf miner attack, but it was an attack on a cultivar that isn't very susceptible to boxwood leaf miner. So winter gem doesn't allow the leaf miner to be very successful. It can't raise its young very well, but because it's making egg-laying punctures into the leaf, you will still get discoloration. So you wouldn't expect this look if you had a planting that was all winter gem, because there wouldn't be a high population of the boxwood leaf miner. But this was from a nursery where there were plants that were loaded with leaf miner adjacent to these relatively resistant plants. So you got this crazy symptom that we'd really never seen before. Another disease that was quite common this year was the Alternaria leaf spot of privet. And Karen may find this unusual, but here on Long Island, everyone plants privet between them and the next person. So it's a really common hedge plant. And the spots are fairly large. They show zones in them, and they can lead to dropping of leaves, which is extremely upsetting. The inoculum clings onto hedges all winter. These are some that Mina gathered for me to use as inoculum this summer. And so you're never really without the opportunity for Alternaria to attack an established planting. These are leaves that were collected just last week, and they're showing the characteristic yellowing that can come with any Alternaria disease, as well as the lesions themselves. And it's not just a matter for privet, that's our major Alternaria host, but we also see Alternaria affecting lilac. This again was a very recent sample with Alternaria leaf spot on lilac. To stop this problem, you'll want to reduce leaf wetness any way you can by things such as spacing the plants better and timing the irrigation. And then there are a number of fungicides that can be used, but they need to be started out on privet in particular early in the season, just as growth begins. The heritage is not labeled for use on privet, but other fungicides are, which are listed here. And if you have a very regimented program, you can control the disease with fungicides if that's needed. Another cousin to that problem is a leaf spot caused by a fungus called Cercospora, and it has long skinny spores. This particular fungus can also go after privet. So one of the things I look for in the lab is which fungus is attacking the privet. The spots don't look that different from one another. We also see Cercospora regularly on hydrangea, and this was the main problem this year. Sometimes it gets really ugly. And again, gee, you wish you could tell just by looking at it. It's quite characteristic for it to have a purple rim like this, but anthracnose can look very similar as well. A relatively new disease to me was the Pseudocercospora leaf spot occurring on the tree lilac, which looks similar except for the spots being a little bit more brown rimmed instead of purple rimmed. And this also leads to defoliation, we found out this year. And here's a lilac, a Syringa vulgaris with that Pseudocercospora leaf spot. And an entire neighborhood was apparently getting defoliated from this problem, which we thought was a really interesting phenomenon. To manage this disease, same basic principles. You want to keep the leaves as dry as you can, and use pretty much the same fungicides for a fungal leaf spot problem. But I just wanted to point out that thiophanate methyl, which is represented here by 3336, is something which does help against Cercospora, but which is not at all helpful when you're trying to control Alternaria. Some of these things have a few quirks like that. So it's good to know which of the leaf spots you're trying to control. Leaf blights included this one, which was unusually common this year on arborvitae. People would think they had arborvitae leaf miner, but they didn't. And the dieback was a lot larger than what we see with things like Pestilotia, which very commonly gets into the tips of arborvitae. We're talking maybe 5 or 6 inches worth of dieback in many cases. And this was associated with Phyllosticta thuji, which can affect not just this plant, but also get onto the Leyland cypress and Chamaecyparis and junipers. So we might be seeing it on more than one plant, really. There are a lot of predisposing factors for it, things like winter injury and drought and plant transplant stress. And I think this year it was the amount of rain on top of all those other things, which are relatively common in their own way. There are a couple of fungicides that are labeled clearly for use against this problem, such as Amancozeb or Spectro. So if you had a problem with this year, it might be worth treating for next year. Anthracnoses are very common leaf diseases that thrive in high moisture. And this oak leaf is showing you a very classic look of an anthracnose lesion. You've got the brown sort of bleeding along the veins. that's the way an anthracnose tends to look. There can be scattered spots, but it's that browning along the veins that's characteristic. And this is characteristic too. This is a shot from a day we were visiting with Sherry Romar in Brooklyn, and you could see these trees all looking really decimated by something. And that was because of the cool, wet spring weather that had fostered sycamore anthracnose. The actual lesions can look very much like what you saw in that oak tree, but the amount of devastation is so high in a sycamore, and in some years like this one, even in a London plane. Here's an anthracnose spotting an ivy. Here's anthracnose on bayberry. Here's anthracnose on hydrangea. There were many different anthracnose diseases that had a good time this year. Here's maple anthracnose taken by Betsy Lamb up in Ithaca. And there were also some spot anthracnoses, which are cousins of anthracnoses that can cause tinier spots as a general rule, and are particularly notorious for messing up the bracts of dogwood. We saw spot anthracnose on roses as well. That has a tan center to the lesion and a purple outside. So realize that not every spot you see on a rose is going to be black spot. I'm running low on time, but I'm just going to go fast here and show you a few more things. Here's a rust on a chrysanthemum. This is the brown rust disease on a cultivar called pumpkin pie, to keep our pumpkin theme going. And the disease that's more of concern is the one that affects many different kinds of chrysanthemum, that's called the chrysanthemum white rust. This is a federally quarantined rust, and I only know of one case, but the fact that I know of one case in a neighboring state to New York means that anyone who is trying to maintain these plants, especially in a production situation, should be using some fungicides to protect the plants. Pear trellis rust is our main tree rust in recent years. It will cause large reddish or orange lesions on the pear leaves, and then it will also do terrible things to its alternate host, which is usually a Juniperus chinensis. Particularly this cultivar, Robusta green, is really prone to it. We see symptoms like this right now. This picture was taken last week in Queens. Swellings occur on the base of the pear leaf, and then they develop further into these funny little acorn-type structures with vertical fissures in them, and spores are released there, which will go to the alternate host, the juniper, and probably none of you are spraying junipers to protect them right now. So that's why this has been such a devastating disease for us. For your rust diseases, again, you're going to want to reduce the leaf wetness, scout for symptoms, and use fungicides such as the strobilurins and the mancozebs. Powdery mildew, you know the look of. Here's a peony. Here's powdery mildew on a dogwood. Unusual symptom, but that's exactly what was going on there. And here's powdery mildew affecting the branches at the end of the tree, which really lets you know that powdery mildews can do different things that are different from what you ordinarily expect. The 9-bark is really prone to this problem, and you can see the amount of inoculum on the underside of the leaf at the right there. And we also even saw black mildew this year, just to show you that crazy things were happening this season. I'm going to stop there just because I have to stop sometime, and I'm watching the clock and it's dinner time, so we don't want to make anybody real hungry. Does anyone have any questions for me?
There are questions, but how many more slides do you have, Margie? Because
I don't know. I have a little beech leaf disease, for example, things like that.
All right. Do you want me to, launch your other poll or no?
Go ahead and do the poll.
All right. So I'm going to launch the other poll, everyone. So get back to your computer. And, you don't have to be correct on these polls. I never look at your answer. I just look to see that I see your name 2 times, 3 times, whatever, how many polls there are.
And I'll take them forward to the picture that shows this so that they can have some satisfaction.
Okay. And Margie, there is time for, both you or Karen to go over a little bit, but we can do a Q&A on any disease, so if folks have questions about other diseases, feel free to put that in the Q&A as well. Because, between the 2 presenters we have on this evening's webinar, we've got a lot of knowledge. All right, so I will end the poll now and share the results.
Okay, so we've got it's split pretty well here about whether nematodes lay eggs or not. So that's cool. All right. So,
Margie, we have someone in the chat saying in all caps, please discuss beech leaf disease.
okay, let's go up to beech leaf disease then.
All right.
They're really determined, okay? So we're used to foliar nematodes occurring on perennials, but the new problem that's a nematode problem is this beech leaf disease caused by Littlelinchus crenati forma specialis
Wait,
mecanii.
Margie, your screen isn't shared anymore.
Oh my goodness, okay, excuse me. All right, okay, so let's just take a moment here. This is what happens when you try to hurry, everything gets all fouled up. Okay, share screen again. I was trying to be so polite and not run over. That was my mistake. Okay, have you got my screen now?
Yep. You just have to put it in the slideshow view.
And amazingly, I hit current slide. Okay, so this is what that disease looks like. If you're off in the forest looking at the American beech, there are galls formed in the leaves by a nematode that is arboreal. It mates and lays eggs within the leaf tissue and multiplies in there. And you have symptoms like this as a result. They're very shallow galls and they cause this sort of striped look on the foliage. Nina and I worked together last year to really chase leaves across the season to see how these symptoms developed. And it was absolutely fascinating that you could see the bands in May that were still there in October, but they didn't multiply. So the symptoms you have in the spring are the symptoms that you have in the fall, and it's all about what happens in the buds over the winter. This is not just a disease of the American beech, it's also a disease of Vegas sylvatica, the European beech. Here you can see, if you have a little imagination, that there are bands that are thicker and darker green in these European beech here, and it can be quite destructive to them as well. Here's a close-up of European beech from a nursery. This picture was taken just this September, and you can see a yellower pattern that's more like what we see in the American beech. The nematodes themselves are long. They look a lot like the ones that we use to help us fight back against insects. And they have their little feeding mouth part called the stylet that they use to feed on the leaf tissues. And they have a really destructive impact on the leaves. This is a picture from Lynn Carta down at Beltsville, who's one of the USDA nematologists working on this. At the left, you see a healthy leaf in cross section with its mesophyll down at the bottom and palisade layer above. And then over at the right, what looks like blue macaroni are nematodes that are twining through the leaf tissue. And that's how it would appear if you cut a slice through a leaf. You also can tell that that leaf tissue is very desiccated and miserable. So it has a lot of negative effect on the leaf when these nematodes are feeding on it. The action is all happening in the buds now. There are not too many nematodes still in those leaves. They've moved to the buds. And here's a wonderful photograph that Mike Gaines got just about 2 weeks ago, showing a female nematode that has been laying eggs. And that's what these little things are here within the bud scales. And they can do that within the leaf galls as well, but it's a pretty amazing thing to see. And we also have some beautiful images from Sandra Jensen in the plant clinic up with Karen. These are the eggs of the Littlelinchus, much magnified. And this is how they look when they're first laid. And then over at the right, you can see the first stage of larva developing inside the egg. And at the left, you see a freshly hatched larva looking pretty lumpy. So these are the things we're trying to learn how to control. Large numbers of nematodes and large numbers of eggs and larvae accumulate in the foliage. And we don't know how to control it yet. Whoever asked for more about this disease probably was hoping I would say how to manage it, but we're working with what is labeled, which is emamectin benzoate as a injection to try to affect the nematode in the plant. And then there's some experimentation skirting around the edges of what can legally be done to use treatments that can be used against mites or against the Phytophthora and see if that happens to have any impact on the nematode. So we're at a time of discovery here. If anyone learns anything or has any bright ideas, I'd love to hear about it. So that's as far as I'd really like to go, other than to show you that the fungi have grown very large this year. This is a giant puffball, and you can turn them into really seasonal items if you really are creative and clever. So,
all right,
now
thanks, Margie.
Okay.
Yeah, I think there are a few in there, but we are starting to encroach a little bit now on Karen's time. So why don't you go ahead and answer one of them. The others, you can always type in your answer. Sorry, I'm always like leaning so close to my computer. I always forget.
Okay.
I undid my video. Anyway, so Margie, yes. So folks, Margie can type in the answer for some of these, and you can look at the answers later. But Bruce McMaster asked an interesting one, I thought.
I don't know anything about the effect of vinegar on nematodes, but it's one of the things that we should consider. We might just be able to change the pH. Maybe a lime treatment would be beneficial. Maybe a vinegar treatment would be beneficial, but we need experimentation on all these things to get a real answer. Right now, nothing has been demonstrated to be effective. Okay. And I will type in answers to the other questions.
Okay. All right. Thank you so much, Margie. I didn't mean to make you rush there.
No, it's
if we
okay.
have more time,
you know,
Margie can always answer those questions at the end as well aloud for folks that have difficulty finding where the Q&A is. I think people who watch the webinar on their phone, it's tricky to get to some of these screens. All right, so now we are going to move on to Karen's presentation, and she's going to give us a really neat update about an early warning system regarding oak wilt. So there you go. Karen's going to share her screen. And I see it. Looks good.
Okay, there we go.
All right. Thank you, Karen.
Thank you. And thank you for inviting me to your webinar series. I'm excited to present this material. I love spending time looking at Marjorie's pictures. So this is a treat for me as well. So I was asked to talk with you today about an early warning system we're kind of working on for oak wilt. And I don't know if you all have a really good background with oak wilt, so I'm just going to take a couple minutes to kind of go over the basics of oak wilt and our brief history with it. I love showing this picture because it's kind of sad to see that this huge oak tree was removed from Greenwood Cemetery because it was found to be infected with oak wilt. And you can probably imagine some of the approaches that we take to minimize the effects of oak wilt have unique challenges in a cemetery. So it doesn't matter where we go, but we really have to think about where we're located, where these trees are, what's doable for management recommendations, one of which is actually trenching, which probably isn't a good idea in a cemetery. So we do have to think about what is the best management recommendation for each individual situation. So let me just give you a few slides on the background. I'm adjusting here. I've got two screens and usually I have three, so I have to adjust a little bit. Okay, so oak wilt's name is Bretziella fagacearum. That recently changed in, I think it was about 3 or 4 years ago. Previously, it was Ceratocystis fagacearum. So you may see it written both ways. Just know it's the same organism. The disease was first discovered in around the 1940s, 42, 44, depending on where you look. And we really don't know what the origin is. Where did it come from before we found it here? So over the years, it built up a range throughout Wisconsin, Michigan, and then slowly moved down to Texas. It was found in Pennsylvania in the 1960s, and then most recently moving down to Texas in the 1990s, and then here in New York State. So let me just show you that little yellow dot right there is where the first case was in New York State. It's in Schenectady County, a town called Glenville. And it was approximately 180 miles from any other known case of oak wilt. So we don't think this was a natural spread. We feel like it was probably a man-made spread because that's quite a jump from any other known cases. And that was in 2008. The folks at DEC, the inspectors, and all of our friends that are landscapers and county extension staff were really looking for this pathogen after we had that first case. So we were out there looking, but we really didn't see it again for about 5 years. And then we had a case in that same neighborhood in 2013. So we had a second case 5 years later. So these are kind of sad pictures, but it just shows what a significant pathogen this really is, and that we don't want it to establish in New York State. So we take it very seriously when we find trees that are infected. So in this case, this was the 2008 neighborhood. Shortly after that identification was made, the team came in, took out about 76 trees out of that neighborhood. They were chopped down, cut up, put into these large piles, eventually trucked out of the area and taken to a burn facility. So what we did when we did this discovery is Sandra Jensen, our diagnostician, was the one who found this pathogen. And typically what we do is we try to isolate from the tissue, and then we'll let it grow out in culture. And then she'll look at that to see if she can identify the fungus that's growing in culture. So she did that, and to her surprise, she found the oak wilt pathogen. We had been looking for this for many years. You saw the map just a minute ago, and you know that it was in Wisconsin and Michigan for many years. They have very similar trees as we do. They have a very similar climate to ours. So we were kind of surprised that it wasn't here, but it wasn't. We were really looking hard. We had samples coming in all the time with the question mark, oak wilt, question mark. But we never identified it until 2008. So when we got that, we wanted to be sure that our morphological ID, looking at that culture plate, was accurate. So we sent our culture off to some experts at Iowa State University, Dr. Harrington's lab. They did a molecular test and then followed that by kind of a second molecular test. They sequenced it. And in both of those tests, it came back to say, yes, Sandra was right in her morphological identification, and it was the oak wilt pathogen. So after those 2008 discoveries, as I mentioned, we were looking for this thing and we just didn't find it anywhere. Samples were coming in on a regular basis. We were not able to isolate them again until 5 years later. And when that did happen 5 years later, the sample was first sent in with the question mark, could this be bacterial leaf scorch? And they are often confused, those two pathogens, and they can be on the same tree. So we did test for the bacterial leaf scorch pathogen, Xylella fastidiosa, and it had a mild positive, but based on the symptoms that the submitter was presenting to Sandra, her spidey senses must have went off because she felt like something else was going on there. So she asked for some additional tissue from that tree that we needed to look for oak wilt. When she did get that, she was able to isolate it again, unfortunately. And we had our second confirmation of the oak wilt pathogen. So the pathogen spread primarily two ways. We feel like in New York State, our primary means of spread is through beetle vectors. The beetles get into these trees through cracks. You can see in the picture there's a large crack that's kind of surrounded by a cut line. The beetles can squirm their way in there. They get the pathogen all over their bodies. They leave and they go to another tree and they introduce the pathogen to the tree because it's kind of hanging on for the ride. We also think man-made spread is common. And unfortunately, this is another case of that firewood being moved. When trees die, people cut them down and they're often used for firewood. People don't understand at times that they could be carrying these harmful pests and pathogens. So it is quite an educational thing that we need to do to let people know this is significant and there are consequences to moving firewood. And this is a perfect example of that. So the second primary way of it spreading is through root grafts. So this picture, you can see it looks like two roots have fused together. Trees can do this through very long distances. It's a great thing for trees to do because they can share resources, they can share food and water, but unfortunately pathogens can spread this way too. And oak wilt, the Bretziella fagacearum is one of those organisms that can move through these root grafts. So when you have an infection pocket, it may start getting bigger and bigger and bigger. And we believe it's because those roots are grafted together and they're sharing the pathogen that way. So how do we get samples in the diagnostic laboratory? Well, we ask for these sections of twigs, usually about 1 inch in diameter, and we also ask for leaf tissue. We want to look at that leaf tissue to see what the symptoms look like. We don't test the leaf tissue. We need those twigs for that. So the leaves are used just to look at the symptom expression. The twigs are used to look for streaking underneath that bark. And then when we see that streaking, we collect that material, and then we're going to do 2 things with it. We're going to use it to test it with the molecular test, and then we also plate. So every sample, we do both of those things. As a diagnostician and working on such a significant pathogen, we want to make sure our answers are accurate. So when we can have more than one test tell us the same thing, it helps us sleep a little bit better at night. So just to show again, this is the leaf symptoms we are looking for. It's kind of a marginal scorch. They may have premature coloration changes. Sometimes the leaves stay green, but they may also turn yellow or brown, and leaves drop suddenly. All colors will be on the ground. So you'll see that intermixing of colors, maybe in July, which is of course way too early for the leaf to drop. We also see this streaking in like a blue-gray smear, where you see that cutout portion on the slide there. That right in the center, that kind of smear is what we're looking for. Other pathogens cause what we call a brown streaking, and that's very different than what this pathogen does. So it does stand out as a different thing. We attempt to make those isolations. So I just wanted to show you what this looks like in culture. We collect that tissue in those little tubes. We put them on these plates of a nutritive medium, something that's going to feed that pathogen. And then as it grows out, it turns very dark and produces spores that we would look at microscopically to identify the pathogen. The challenge with this organism is that it is very devastating when it's in the tree, and it can cause a decline very quickly. However, when we try to take it out of that material, it's not as strong, and it's quite a challenge to try to get it in culture. So we spend a lot of time and a lot of plates trying to get that out of the tree if it is infected. And then we incorporated this recently, and I'll talk about that in a minute. But a few years ago, because we had more than one case, we decided we needed to be doing this molecular test as well, because we were getting so many more samples, and we wanted to make sure that we were doing everything we could to identify these infections as early as possible. So we had a better chance of possibly eradicating it. So we incorporated the double nested PCR into our protocols. And this just shows here one of the commercial kits that we buy to do the DNA extraction. And then the red machine there is our conventional PCR machine, which means we have to run it out on a gel. And that picture there of the gel, you can see a number of samples lined up there. And then at the end of that run is the positive control. So you have a line that's clear, and then the positive control next to that. And you can see these are all lining up quite nicely to that positive control.
Karen, I'm going to go ahead and launch the poll, one of your polls. So folks, go ahead and get ready for the poll that's about to pop up on your screen. What do researchers believe caused the initial introduction of the pathogen into New York State? People planting infected nursery stock, infected firewood movement, root grafting of infected trees to healthy trees, wind blowns, or transmission. So I'll leave it up there for a few seconds, and then I'll share the results.
Great. See, this tests us too, on how good a teacher we are.
Well, I think you're both excellent teachers.
You're too kind.
All right, it looks like 40 seconds are up, so I will end the poll. Anybody? For folks that are having trouble seeing the poll, go ahead and just type in chat that you're paying attention. You don't necessarily have to do the poll. If you can, it's just an easier way for me to keep track that you're paying attention. All right, so you should be able to see the results.
Here we go.
So most people chose infected firewood movement and then planting infected nursery stock.
Mhm.
All right, so I will stop that again. If you missed the poll, please just go into chat and say that you are paying attention. Thank you. All right, back to you, Karen.
Yes, the answer was the firewood. Yes, we do think it was firewood. It could be infected nursery stock. We don't know that it wasn't. But however, with this pathogen, it tends to infect older trees. We have found some that are younger, but in most cases, trees are probably at least 10 years old or a little bit older. So that's kind of just a general observation. It doesn't mean it can't infect young trees too. So I wanted to put this slide in my presentation tonight because we had a case recently that brought this to light, and I wanted to make sure I pass this on to all of you. Please be very careful when you do choose a diagnostic laboratory to send your samples to. You want to make sure that you're choosing a facility that has really good lab practices. You don't want to make costly decisions of your time, money, maybe introducing pesticides to the environment based on answers that aren't really valid. So it really does, it really is beneficial for the user to take the time to look for a facility that they have a lot of confidence in, because you are going to be making significant decisions off of that answer. And we have colleagues all over the country that can help you. We have great colleagues here at Cornell University that can help you. So I just wanted to put that in here to make sure I brought it up, but also to give you our contact information too. So that's my picture there holding up a plate. That's actually the pathogen we're talking about tonight. And then there's a nice picture of Sandra. In case you don't know Sandra's face, now you can see her beautiful face as well. So basically, I work on mainly the survey symptom or the survey samples in the laboratory. So the oak wilt, sudden oak death, and a few others. And Sandra works on all those random samples that come in. So she has a very broad knowledge of pathogens that affect everything from annuals to turf grasses and fruit trees, vegetables, ornamental plants, everything. So, okay, so I want to share with you a few other things. So I don't want to hold up too long on this point, but I want to tell you a little bit more of the story. So I pursued a specialty crop block grant after we had our second case now, and thought maybe we should be looking for this a little bit more, and that maybe we should be doing that molecular testing. And by requesting this grant, it would allow us to test that protocol. I could implement it into the laboratory, get a number of samples to make sure that it was working properly, and have that as a great resource for us in the clinic. So we luckily got that grant, and it began in the summer of 2015. So to this point, we just had the 2 positives in the Glenville area, and we received about 22 samples that year. And of course, it's always that last sample of the year. The very last one gave us a suspect result. And we couldn't confirm that that's what it was, but it was highly suspect. So we did some work over the winter and early into the spring, and we were able to confirm it that following spring. And unfortunately, we had a few other positives as well in 2016. So let me show you a map of New York State. So that little blue dot there with Glenville written above it is that original find, the 2008 and 2013 finds in that neighborhood. And then in 2016, that suspect case was from Long Island, the Central Islip area. And you can see we had a few other positives from Long Island as well as Brooklyn. And then a big surprise happened a couple weeks after we had these positives. Canandaigua popped up. So take a look at the map. They're all over the place, right? We've got pretty large distances between these sites. Just like we were talking about from the Pennsylvania closest known site to Glenville, these sites are about 180 miles apart as well. It's about 183 miles from Glenville to Brooklyn and 184 from Glenville to Canandaigua. And then from Brooklyn to Canandaigua was a little over that 280 miles. So they're very spread out. Again, probably man-made movement of the pathogen here. Makes the most sense to us that that's probably the case. Just want to show you a quick update of Long Island so you can see it just a little bit better. Down there where Queens County is the Brooklyn, and then Central Islip, and then that other one near the Babylon is actually West Islip. And then we had one near Riverhead and Southwold. So to give you a quick review of what we had since that time, we really ramped up the number of samples we were testing in 2017 and did 144 samples that time. And we actually had a unique case. It was the first time we had a culture or morphological positive without having a PCR positive. Prior to that, we've had a number of PCR positives that we couldn't get cultures on. And that didn't surprise us because we know it's a little difficult. It's a little ornery and it doesn't want to grow in culture all the time. But to have it this way was a little bit more of a surprise to us. So it just drove home the fact that us doing both of these types of tests, both of these procedures is really important, and we should continue to do that. So let me show you the map now. Those two new sites in 2017 were back in the Glenville area, unfortunately. One was about 3 miles from that original site, and another was about 7 miles from that original site. So this is, the last case there was 2013. Now we're 4 years later, 2017, and we got a couple more sites showing up. So that was unfortunate, but our DEC jumped right on top of it and did the eradication efforts that they felt were right for those cases. In 2018, we did about 81 samples and we found 3 new positives. Let me just jump right to the map so it's a little easier to see. We had a couple more in that Glenville area right on top of those previous year positives, but then we had a couple more in Canandaigua. So again, those were found in 2017 and now we're in 2016 and now we're in 2018, 2 years later, we're seeing some spread. So I'm just going to finish this up quickly, show you some of these maps quick. And I just wanted to show you one more thing on the canine project that we were doing. But in 2019, we decided, could we test, well, we actually asked the question, could we test the beetles? If we trapped beetles, could we find the pathogen on the beetle body before we found infected trees? So we tried it out. We put together experimental design and we started testing beetles. And we had 9 positives that year, 2 of which were tree samples, but 7 that were actually beetle positives. So here's the map that shows those beetle positives as well. The purple dots there near Canandaigua were the 2 other tissue samples. Again, now we're spreading a little bit and we're actually into a new county. And then the bees, the little bees here, and I think I actually put circles around everything, so it's a little easier to see. But in Canandaigua, in Glenville, down in Brooklyn, and out in Riverhead, we had beetle positives. In 2020, we did a lot of beetles. I spent many nights and weekends processing stinky beetles. We didn't have any tissue positives, but our numbers were way down. This is the year of COVID. And we weren't allowed to travel as much. The DEC folks weren't allowed to travel as much. So we really focused on trapping beetles and testing those. We had 20 positive beetles that year. So you can see these are in the blue bees. And again, I did circle those so they stand out a little bit. Most of them were south of Canandaigua, which is really interesting, and one over in Glenville. So the efforts this year were really expanded in those counties to see if there were any infected trees and if we are still getting beetles showing up with those infections. So let me just tell you about this really cool project quickly. I'm going to run through it really fast so I can answer some of your questions. A couple of years ago, we put together a proof of concept study to see if dogs could find the oak wilt pathogen. Dogs are really good at finding other things like fruit and insects and weeds. And there is some work with plant pathogens in the past, even some viruses that they've been trained to find. So we wanted to know if it would work with oak wilt. So we worked together with a great group of people, these folks out west called the Working Dogs for Conservation. They have these dogs that are trained on all kinds of organisms. Go to their website. It's a really fun read, but you can see here Atlas, who is a Labrador Retriever. She's trained to find shark fin. So if that's an illegal trade item, so she works, on the borders and in customs and finds that. Dogs have been trained to find species that they feel might be endangered or maybe even extinct, but the dogs might be able to find them and let us know if they are still here with us. So it's a really great program. And we worked with Amy Hurt there. She was one of the co-founders and she was so helpful. Josh Basie was the dog handler. He was hired to do a specific project for this. And he trained Dia just for this project to work with the spotted lanternfly, a weed, Scotch broom, and the oak wilt. And Fagin was his human rescue dog. So this was a dog that could find if someone was lost in the woods. He was trained to find people. So he also trained him alongside Dia, which was really fun. So they originally worked on the spotted lanternfly. That was their primary project, but they also wanted a weed and a pathogen. So they added Scotch broom for the weed and oak wilt for the pathogen. This is a little grainy picture of Rob Cole, but he is our DEC representative and he's in charge of the oak wilt program. And Kelsey McLaughlin is right behind him there or below him, showing some pictures of the work that they've done out in the field. They put out these great posters to tell people why they're cutting down these trees. And when we did the training here, you can see Dia in the background. We're setting up a sniff site for her. The dogs took on really quickly. It was amazing. We would put in these containers here just a glass vial with agar in it, and then a glass vial with agar and the pathogen in it. So they weren't alerting on just the glass vial or just the agar. Within about 10 minutes, they had it down. It was beyond remarkable. So here you can see Dia sticking her nose right in there to get a really good sniff of that organism. And if they hit it, they would sit immediately and he would reward them for that sit every time they got it right. And like I said, within about 10 minutes, they were pretty good at it. We started hiding things around the room, up by windows. They were really, really good. We went out to a large livestock barn and we inoculated some actual twigs and put them lined up here. You can see, here's a little close up of that. And then took the dogs just down that line. And again, they got it right every time. It was really amazing. So here's just some of our setups that we did outside to do some additional training with them. You could see we could really hide some of these twigs outside when we took them out there. So they had to really be working for it. And here they are out in a big field out behind campus looking for our little hidden treasures. And we even hid them in this huge pile of bales of straw that was sitting there for a couple of years. And that's Fagin up there, and he actually found it. We put these little canisters up in trees. And look at Dia, she's on her hind legs sniffing, looking for that organism. It was quite impressive. Hid some in wood piles. So, it's really hard for them to see that. They were really finding it with their nose. So this is actually a site that had some positives, and Dia had alerted on the stump of the tree that was positive in that site. So it was just a really great project. We hope to do more of this work because I think the dogs could help us narrow down sites that we should be looking at more closely. It was a lot of fun. We're trying to get some funding to do some additional work, and hopefully we can do that. So that'll wrap me up. I'm sorry I went over a little bit, and I just want to take a few minutes to answer any of the questions you might have.
No, it's fine. You didn't go over it all. I'm going to launch the other poll. So folks get ready. And again, if you can't access the poll, then just go right ahead and comment in chat that you're paying attention. So I'm about to launch it. You should be able to see it on your screen now. Who were the stars of the Oak Wilt Canine Proof of Concept Study? And again, if you get it wrong, it's no big deal.
Right. Yeah, some of it might be your opinion, actually. So,
Yeah.
who you feel the stars are?
I do have a question for you, Karen, regarding the dogs. So how do you know that they weren't just smelling like their owner's scent, or like whoever's scent had placed down the object?
That's a really good question. So what we would do is mix it up quite a bit.
Okay.
So we would have different people place the items. Sometimes Josh would do it. Josh didn't usually do it very often, but Amy would do it, or I would do it, or we actually had some other people hanging out with us to check it out and take pictures. So we would ask them to do it at various times. But you're right, the dogs are really, really smart. So if you're not a good trainer for this specific task, you can screw those dogs up, and you can have them alerting on the wrong thing, such as the glass vial or the metal tin.
Yeah.
You just use one thing over and over again for that training, or it could be the wood, right? If you're using inoculated wood, you might be alerting on the oak instead.
Yeah.
So it's a matter of training them very different ways, and they need to have ongoing training too.
Okay, very
so
cool.
Keep going back to it. It is very cool. Yes.
All right, so there's the answer there. The one that was most picked, Dia and Fagan. So I will stop the share and X out of that. And it looks like there's a couple of questions in the Q&A.
Let me stop my share, but before I do,
you can leave your share up, Karen. You know, just in case there's questions that refer to it, feel free to leave it up.
Okay, so we have those be on the lookout cards, which is on this slide. We have a number of these postcards. We can print a lot more, but if you are all out and about, if you're a landscaper and you want some just to hand out to people to show them, please let us know. We will definitely share as many as you want. We sent a number of these out to county offices years ago, but anybody who wants any of these, please let us know. We want to get the word out there and have people helping us look, because in fact, the first cases that we found were because people were out there looking for it. And in 2016, we put these out before we started doing that survey. And actually both cases, the one from Long Island and the one from Long Island and the one from Canandaigua, said that they had seen some of our materials. And that's why they thought it might be a case of oak wilt.
No, that's great. I was handing these out at our conferences, and I think I still have some on hand, but I'll always take more, Karen.
Sure, absolutely. Let me know how many you want and we'll send them your way.
All right, I'll shoot you an email. Thank you.
Sure. So one of the questions was regarding oak wilt, was the equipment used for taking down the infected trees sterilized before leaving the site? Yes, they had very strict rules of how they do that process. What materials or what tools they could use on site, where the materials went after it left that site. So they actually worked with a lot of the states in the central state area because they had decades of experience with removing trees. So they've talked with the departments of agriculture out there and learned from them what worked best. You know, what do we have to do to ensure we're sterilizing, you know, our equipment, our tools, all that. Now, one of the things that really stood out from the researchers, what the researchers told us, was that it needs the beetle to introduce it to a tree. So if you did have a pile of mulch sitting there and it was all chopped up, and say you were just leaving that pile, like maybe covering it with a tarp, my question always was, well, couldn't the pathogen get out of there very easily? And maybe, but this isn't a classic windborne pathogen. So some of those windborne pathogens, we know they produce these structures that just a little air current can pick them up and they can travel miles down the road and infect new plants. The late blight fungus that infects tomato and potato, right? Bill Fry told me, the researcher who worked on, the famous Dr. Bill Fry, who worked on late blight for many years, had done studies, and he learned that it could travel 7 miles from an infected site to someone else's garden 7 miles away. So this pathogen isn't known to be an airborne type of pathogen. It's found internally in the tree, and the beetles get in there, feed on it, and then move to the next site. So it's kind of relying on that beetle to get it to where it needs to be, into that vascular system of the tree. But they are hyper vigilant about making sure it's not us spreading it anymore, right? So we're educating people on firewood, telling people if they do cut down trees, they need to keep that wood, you know, close to home, and what other steps can be taken to protect those sites. Removing the bark is a big thing. So I know they recommended that in a lot of cases too. Just removing that bark exposes the pathogen to light, sun, the elements, and it can dry out and die more quickly than if the bark stays intact. Let's see, the next one was, can Karen stress the no prune in summer? Yes. Yes, the no prune. So if you go to some of these other states, you may see posters or billboards up that say, don't prune in May and June. So they have that catchy saying, because of oak wilt, don't prune in May and June. Well, here in New York State, they recommend to even extend that a little bit. So they're recommending more like March to September should be the no prune time. And the reason for that is because the beetles are out there, the pathogen is sporulating. So it's an ideal time for that spread to happen. And if you make a natural cut or, you know, an actual wound with your pruning shears, it's kind of a flag being waved around to the beetles to say, hey, here's a great place to come enter this tree. Jenny Jeswick did some study on this where she had graduate students watching in a known infected area. They would make a wound cut and they would see how long it would take for those beetles to show up on that wound. And in one case, it was as quick as 10 minutes. So that no pruning thing in the summer, sometimes people have to. Sometimes it's a wound that might be from a winter, or a summer storm that caused a wound, not a prune. But if you don't have to, we recommend you don't prune during the summer. If you have to prune, the best time to do it is in the winter. It's probably not bad from November to, you know, whenever the snow falls. But the best time really is January, February, and March. And if it's an early spring and starts warming up in March, I would wait so that you're not doing it from March to probably September. So thank you for bringing that up, because that is a really critical point. And I know after we've done educational sessions, Marjorie talks about oak wilt a lot too in her presentations to get that word out there. And I've had people call me after seeing one of our presentations and say, I work for a town and I've got a pruning crew coming in here in a week. I don't know what to do. What do you think? And a number of them have canceled that. And I'm sure it was costly to do that and have to put that on hold, but it's really worth protecting the trees. So if you don't have to prune, don't. But if you do, they recommend just using just plain spray paint or some kind of wound dressing that covers it so the beetles can't get into that wound. So I know that's a big debate too. Do you use wound dressing or not? Lots of people have strong feelings one way or the other. In the case of oak wilt, yes, use the wound dressings. So let's see, and we saw the foliage of a batch of swamp white oaks quickly turn brown and fall off within 2 weeks' time. I looked at the roots thinking it was rot, but the roots looked healthy. Could it have been oak wilt? Possibly. One of the things that we have been trying to convey to people is that white oaks can become infected. Some of the literature out there, some of the advertisements out there say it's just red oaks. And that's not the case. Red oaks are much more susceptible than white oaks, but white oaks can become infected. They're usually, they don't go down as quickly. The infection doesn't progress as quickly. So it may be a matter of years for the decline in white oaks, where for the red oaks, it may be a matter of weeks. But white oaks can become infected. One of our first cases, like probably the first 3 or 4 that we had, one of them was a white oak. And when you see the leaves drop that quickly, that's a really characteristic symptom of oak wilt. So there's a number of things. We look for that leaf scorch. We look for all colors of leaves dropping quickly. And we look for a quick dying of the tree or a quick dying of a branch. It could be just like a flagging branch, kind of like Dutch elm disease will display. So that one branch going down quickly is a really good indication of a good candidate for testing, I would say, for those trees. Do we have time for one more?
Yes, yeah, we have 10 minutes more.
Okay, so a client has lost a few trees on her property due to oak wilt. Is there anything that can be done to prevent it from spreading to the rest of her oaks? What they do in other areas is remove the trees that they know are infected as quickly as possible. They grind out the stumps because that pathogen can remain in the stumps. And then they do the trenching. And if you look online, you can see they've got some nice descriptions, some diagrams that show how to do trenching. And typically what I've seen is, say you have 3 trees in the center of a field that are infected, they might go out like 50 feet or 100 feet from that infection site and trench all the way around those. And then do a second one, maybe another 50 or 100 feet out, and do another trenching. So what they're doing is, in case that root grafting has moved the pathogen to that first circle, to those first trees that are around the infection site, that second trenching hopefully will break it from going on even further. So I've seen this trenching done. The buildings shake for sure because they use these 5-foot long vibratory plows. That's just like a big tooth that goes up and down. It breaks those root grafts so that if the pathogen is right back on site 1, it can't transmit that way. So I think quick eradication is really important. Trying to get as much of the material as possible. And then if you are trying to protect a site, the trenching is probably the best known method. There are tree protections that can be done. Individual trees, if you have like a specimen tree you're trying to protect, I know especially down in Texas, they use a lot of treatments to protect the tree. I believe that it doesn't last forever. It's probably something that needs to be done frequently. And I thought they told us when we were in Texas, Marjorie and I went there a few years ago to see their situation. I think they told us that trees could really only handle 2 treatments. So you didn't want to do it too soon because you wanted to kind of save those 2 treatments for when you really needed them. But I think for us here, probably trenching, and it depends on the site too, right? If it's a real rocky site, trenching might not be an option. So you have to look at the site to decide what the best way of protecting the trees is. Any other questions? We got one more. Are the trenches treated with anything? I don't believe they are treated with anything. What I have seen them do is they'll either do this vibratory plow that just kind of makes a straight line, or use like a small backhoe and make a trench. And I have seen them put some kind of, it's not like landscape fabric, it might be even thicker than that. Like it's a barrier treatment to slow the grasping of those grafts. So the grafts don't reform in a quick turnaround time. They do put some kind of material in there, like a really heavy plastic or like a steel mesh plastic combo or something like that, that will go into that trench. They fill it back in, but they think that slows down the regrafting of the roots.
All right, I think you covered it all, Karen. Margie,
Okay.
do you have anything else you would like to add? We have about 5 minutes more.
No, but if anyone has a question, I'll be happy to take it. So.
All right. Could one of you address when was the last infection of oak wilt on Long Island? Was it 2018?
The last was the beetle that Karen found.
Yes.
And I was excited you just mentioned that there was one in Riverhead. I didn't know that before.
Yes.
Very
Oh, okay,
exciting.
I missed
Maybe
that.
from my
All
own
right.
backyard.
Right. So
Yikes. Okay.
we this past year, I was quite shocked to see how they really gathered together. So those little bees that were on our map, there's 4 little bees kind of south of Canandaigua down into the counties just south of it, of Yates County and Ontario County. And they were all 4 different state forests. And at the time that I was doing the testing, it was, just randomly doing the testing and getting the positives. I don't check where it is when I'm doing the testing. It wasn't until after the fact. And then all of a sudden I'm looking at the map and saying, oh my gosh, they're all right next to each other. That was really interesting to see that line up that way. Yeah. It's a little fruit fly flying around my face here.
I can't see it.
No,
All right, well, I think we've addressed everyone's questions then, and I want to thank you both for taking the time to update us on the various diseases in oak wilt. I will send a follow-up email, so if you have something that you want me to share with folks, please send it my way. I'll send out the email on Wednesday. Karen, someone did say, how can I contact you to get those postcards? So I'll include your contact information along with Margie's in the email, so they can reach out to you. Yeah.
perfect.
Another thing that you might want to do when you write back to people is to include the information on how to subscribe to Branching Out, which I noticed you had in your introductory materials, because now's the time folks can sign up for next season's Branching Out at a slight discount, and then they won't have to remember next spring when they get real busy.
Okay, I have been providing one of the emails. I provided the link to Branching Out.
Good.
Because Dan had discussed it last week. So I don't forget which email it is now, but I'll definitely do it again.
Great, thank
I've
you.
put this on my little note for next week. Okay, all right. I think we're all set. So good night, everyone. And just know that I'm working on the continuing education credits. October 14th is done. So if you haven't received your landscape architect certificate, reach out to me. because you definitely should have by now. And I email the ISA folks and CNLP, so you should see those posted on your accounts sooner than later. But I'm working on the DEC credits now for October 21st. So just give me some time. Hopefully I'll be done with all of those credits next week. And these credits, the October 28th credits, will be in a couple of weeks. So thanks, everyone. Good night.
Thank you.
All right. Thanks,
Good
Karen.
night.
Thanks, Margie. Bye-bye.
Thank you.
So long.